Live right now. Welcome everyone to the PGC Worldwide Lab Meeting. This is being recorded and we'll post the recording later for people to review who couldn't make this time or call or who wants to look at stuff as well. It's a deep pleasure to introduce to you Charlie Gersbach. Charlie is at Duke and he's one of the world's experts on CRISPR. I've been working with him for a couple of years now. And it's been uniformly fantastic, especially given all the complexity and novelty and interest that Charlie has in developing tools that can be used in the business of the PGC. So I will turn things over to Charlie and he will take it from there. I believe he's okay with taking questions at any point throughout the talk. There's a Q&A feature or a chat feature with Zoom. If you use either, we'll tee up the questions. And I believe he will also leave time at the end to take questions from people. So Charlie, I'm really, really pleased you've done this and I look forward to your talk. Thank you.
Yeah, thanks, Patrick, and thanks for the nice introduction and the opportunity to talk about some of the work that we've been doing. And what I've decided to focus on is really this area of how we're using epigenome editing. And I'll talk about some of the tools that we've been developing and how we've been characterizing them. And then at the end, I'll talk about how we're using them to kind of characterize non-coding genome and follow up on GWAS hits and the types of things that I think are most interesting to this audience. And as Patrick mentioned, I'm happy to take questions along the way, or there'll be time at the end, I hope, if we get to move along. So I'm required to show some disclosures for relationships with some companies that I have when I present this work. And so my lab at Duke, we've been here for about 8.5 years now, and we've been working with these genome engineering technologies quite broadly. So I started my lab working with zinc finger proteins and kind of earlier genome engineering and gene targeting tools before the CRISPR systems. Originally with zinc finger proteins and then with tails and tail ends. We've also done some work with meganucleases for targeting, but more recently, we've really been focused on using the CRISPR system for all of the reasons that everyone's heard about, how easy and fast and cheap it is to work with. And these are some of the areas that we've been using it in. Kind of genome editing, as most people are familiar with using the CRISPR system for, so cutting DNA and introducing DNA sequence changes. And one of the areas that we're using that is to correct mutations that cause rare inherited genetic disorders like muscular dystrophy. But what I'm going to focus on today will be the stuff over on the right here, where we've been using it not to cut DNA, but to control gene expression and change epigenetic marks. And I'll highlight some of the ways that we've used it, for example, in controlling cell fate specification and decisions. I think it's a good example of how the technologies can be used. And then I'll talk at the end about how we're using it for screening non-coding variants. So the idea of epigenome editing in general is very similar to gene editing, in which you have some type of DNA binding domain, like I'm showing here in the middle, that can be targeted to any DNA sequence. And as I mentioned, we've used a variety of technologies for that: zinc fingers, tails, CRISPR, et cetera. But instead of cutting DNA and changing DNA sequence, like people can typically think of with gene editing tools or CRISPR, we're not cutting the DNA, but rather either directly attaching or recruiting what we call the epigenome editing effector. And this could be something that modulates epigenetics or gene regulation in any way. So something that activates genes, represses genes, or modulates a specific epigenetic mark. And so we've been developing these tools and using them for a lot of different applications that we're really excited about. Starting over here on the right, one of the ways we're using it is to better understand mechanisms of epigenetic regulation. So while there's been, maybe dozens of different epigenetic marks, different types of changes to histones and DNA methylation states that have been described, almost all of these have been correlated with changes to gene expression states. So particular histone marks that are usually present when genes are off or usually present when genes are on. But the actual cause and effect mechanisms are very poorly understood. So it's not clear whether a particular histone methylation is there because the gene is repressed, or the gene is repressed because of the histone methylation. And so what we've been doing is developing tools where, rather than just characterizing these epigenetic states, we can go in and change them directly and then see what the downstream effect is on gene expression. And so that's one way to use these tools to better understand causative mechanisms of epigenetics. Going clockwise, We're also using these for high throughput screens of cell phenotypes, but also using this to typically when people think about CRISPR screens, they think about these high throughput knockout screens where you're knocking out coding genes. But we want to use these tools for screening the non-coding genome and different epigenetic states and how that leads to changes in gene expression and cell phenotype. And I'll talk about that more at the end. We also have some projects in the area of synthetic biology. We're making gene circuits out of these tools. We are also, I'll show a slide or two where we're trying to use these types of tools for modulating targets that might be interesting for gene therapy applications, and then also for understanding cell fate specification and creating new cell models of disease and also for drug screening. And I think I'll give an example about that as well. So I mentioned that DNA binding domain that you can use to target the any site in the genome. It could be a variety of things. It could be, like a zinc finger protein or a tail, but we've mostly been using the CRISPR-Cas9 system, like many others, for the reasons I mentioned. And as probably many of you are familiar, the CRISPR system in its engineered form, as it's used for genome engineering or gene editing, has two components: this Cas9 enzyme that I'm showing here in yellow, and is the nuclease enzyme, and it forms a complex with a short RNA called about 100 nucleotide RNA called the guide RNA, and the enzyme in yellow forms a complex with the guide RNA in red via a structure in the guide RNA at the 3' end, and then that complex goes and finds the target site in the genome. First, based on this PAM sequence, it's an acronym for protospacer adjacent motif, and in this case, for this CRISPR system, it's any NGG sequence, where N is any nucleotide, and so you see that here in the middle. But then once it finds one of those sites, it then looks for complementarity between the guide RNA and the genome, and that's shown here as this the red guide RNA is going in and annealing and forming an R loop with the DNA target DNA strand shown in green. And once that complementarity occurs and hybridization, then there's a conformational change to the Cas9 enzyme where it cuts DNA, and that's how this system is normally used for gene editing. So this system of CRISPR-Cas9 for gene editing is sometimes called the molecular scissors. So it'll go in and cut DNA, and then typically you would take advantage of after cutting the DNA, take advantage of DNA repair mechanisms to incorporate different types of sequence changes that you're interested in. But it turns out, as some of you may know, that there's another version of the system where it's called the broken scissors, and this is what we use for epigenome editing. And in this case, it turns out that with this Cas9 enzyme, you can inactivate the cutting function or break the scissors by making 2 point mutations to the enzyme, one mutation to each of the nuclease domains that cuts each of the DNA strands, and that inactivates its cutting function. But it'll still bind to the guide RNA, and it'll still go find its target site in the genome. But at least in eukaryotic cells, it typically will just sit there and won't really do anything. But if you attach or recruit this epigenetic modulator domain that, as I mentioned earlier, changes gene expression or epigenetics, then you can take advantage of this protein scaffold that you can send to any site in the genome to modulate those epigenetic sites at those or epigenetic states at those particular sites. And so this kind of broken scissors is what we're using for these epigenome editing studies. And there's been many labs that have worked on developing these tools. This inactivated Cas9 enzyme we call dead Cas9 or dCas9 for short. And one of the first examples was from Stanley Chi and his collaborators at UCSF a couple years ago, where they attached this CRAB domain to dead Cas9 to make a targeted repressor that they called CRISPR interference or CRISPRI. And this CRAB domain had been used for a long time to silence different target genes. It's actually a domain that's conserved in more than 400 different human transcription factors that are involved in gene repression. And it acts by recruiting HP1 and other heterochromatin-forming proteins. And they showed in the study that when they targeted it to gene promoters, they could silence those gene promoters. And that built on many years of previous work using CRAB domains fused to zinc finger proteins and tails showing targeted repression. So even after this original study, we still had a couple of questions remaining. And One was how specific is this targeted gene repression? Of course, almost any conversation about CRISPR involves discussing concerns about specificity and where else these tools might be acting in the genome besides the intended target site. Another question that we had is how far does this targeted repression spread? We had actually failed to get funding for some of this work for many years because there were reviewers had concerns that even though we might induce heterochromatin at a specific site in the genome, if there was linear spreading of that heterochromatin formation across the DNA, then it wouldn't lead to a very specific effect. So we needed to know how far does that heterochromatin formation spread. And then the original study shown here showed that they could target gene promoters, but we wanted to understand to what extent this tool could be used to control enhancers and other distal regulatory elements. So, and of course, the reason why we're interested in enhancers is because their role in controlling, binding different transcription factors, looping onto promoters, and controlling gene expression, as probably many in this audience are familiar. And so the model system that we used to kind of characterize this tool was the globin locus control region. And we use this because it's probably the most well-studied locus control region in the human genome. And in this case, there's 5 hypersensitive sites, HS1 through 5, shown here in yellow, that loop over onto the different globin genes shown in green. And this looping happens, the nearest gene is about 10 kilobases away, and the beta globin, the furthest gene, is about 50 kilobases away. And these genes, epsilon, gamma, delta, and beta, get differentially regulated during development. And so HS2 will loop over onto each of these promoters to activate target gene expression. And so we wanted to know, can we go in and use this CRISPRI or Cas9 crab fusion protein to repress HS2 and inactivate its function in controlling the globin genes. And so for this, we used the K562 cells, so a human erythroid cell line that actually expresses all of these globin genes. So it's a good model system for testing regulation of an enhancer. So we showed that if we made a guide RNA that was targeted to HS2 that and used the Cas9 crab tool to target that enhancer, that we would get corresponding decreases to expression of these different globin genes. But to understand the specificity of regulation, we used RNA-seq, and that's shown here. And we did a number of different comparisons and controls and looked at a couple of different guide RNAs, but I'll just highlight some of the results. In this result, or in this slide, I'm showing the comparison is with or without the guide RNA, and plotting, of course, fold change in expression relative, again, with or without the guide RNA, and across the x-axis, we're looking at the level of expression of each of these genes. And each dot represents a different transcript in the human genome, and everything in red is significant after multiple hypothesis testing, and the dots in blue are just pointed out for reference. And what we were really happy to see when we targeted the Cas9 crab fusion protein to HS2 with a single guide RNA was that the only genes that were significantly repressed across the genome were these different globin genes that are present in that locus. So that was nice because it showed that we could go and target one enhancer, and the corresponding effect on gene expression would be only on the known target genes of that enhancer. But of course, that doesn't tell us where the dead Cas9 crab might be acting. So it may be binding all over the genome, but it only happens to be changing gene expression in these cells when it lands in the HS2 enhancer. So to better understand the interactions of the Cas9 crab with the genome, we did ChIP-seq for the HA epitope that we had included on the Cas9 crab fusion protein. And if you look across the top, this shows the DNase-Seq data for these K562 cells. And you see there's open peaks at each of the globin promoters, as you would expect. And then on the right-hand side, you see these two guide RNAs, CR10 and CR4, that we had targeted to HS2. And so the track right below that shows that if we deliver Cas9 crab alone without any guide RNA, then there's no ChIP-seq signal at that site. But when we include the guide RNA, either with or without, with Cas9, with or without the crab domain, in both cases, we get a nice crisp peak at the target site in the genome. And then if we look down below, genome-wide, we see this is looking at 300 base pair windows across the genome looking for ChIP-seq signal. And we see the only thing that stands out in multiple biological replicates is binding at that HS2 target. So this was surprising to us that there was this specific, but was really pleasantly surprised that the only robust and reproducible target site across the genome was the intended target site at HS2. Now, if it's binding only at this site and the CRAB domain is doing its job, then it should be recruiting heterochromatin and forming repressive marks. And so to better take a closer look at that, we did ChIP-seq for H3K9 trimethylation. That is a repressive mark that should be forming in response to this CRAB domain. And again, looking at the tracks at the top, we see now we do see enrichment for H3K9 trimethyl, but only when we include the CRAB domain. So that's good news. But also we only see it at we only see enrichment for this histone mark at HS2, and then the two neighboring hypersensitive sites. And this is interesting for a couple of reasons. One is that we know these neighboring hypersensitive sites interact with each other through looping of the genome. And so we see that the effects of CRAB can spread to these sites that it's looping to. But it's also, those peaks at these other sites are discrete peaks. So it implies that enrichment at those other sites nearby HS2 is happening through 3D looping as opposed to linear spreading across the genome. And that helps to resolve some of our earlier concerns that by really tightly tethering CRAB domain to the genome at a single site that we might get heterochromatin spreading over large regions. This, in fact, implies that we can go in and modulate a specific hypersensitive site with some additional effects potentially on the other hypersensitive sites that we know that it's looping onto. And then, of course, down below, when we look at genome-wide, where the signal is, the biggest fold change, we're at HS2, and then this other site that it's looping onto. There was a third significant change that was at another chromosome at an off-target site. And we actually verified that this looks like a bona fide off-target site because when we looked at the DNA sequence under that ChIP-seq peak, we found what looked like a pseudo-guide RNA target site. Obviously, we didn't see that in the ChIP-seq for DEAD-Cas9 that I showed on the previous slide, which may just imply that the chip for H3K9 trimethyl is more sensitive. But nevertheless, you can see that we're very clearly getting highly specific changes to a particular epigenetic mark genome-wide in this system. And then finally, if we're actually inducing these repressive histone marks, then we should also be closing up the chromatin at that site and decreasing DNase hypersensitivity. And so with Greg Crawford at Duke, we looked at this more carefully using DNase-Seq. And what we see here is the most significant sites genome-wide that are changing their chromatin accessibility in response to this targeting is in fact at the HS2 and the neighboring HS3 site. Another way that we looked at this was this volcano plot, where instead of plotting full change versus signal, we're plotting p-value versus full change. We see all the full changes are negative because chromatin accessibility is decreasing as we close up these targets. And then we thought that it was interesting that the most significant sites, again, are starting with HS3 and then HS2, so these two neighboring hypersensitive sites. And then the next most significant changes are the distant promoters that these enhancers are looping onto. And so this implies that when we are repressing and closing up these enhancers, that we get corresponding decreases in chromatin accessibility and silencing of those distant promoters that those enhancers loop over onto. And so, and then finally, that was all with cells and culture. I wanted to give an example of how we've actually then moved to using these tools in vivo in animal models. For this, we started with doing this in the liver as a model system with a model gene target, PCSK9, that is known to be involved in LDL receptor recycling. And it's well known that repressing or inhibiting PCSK9 will lead to decreases in LDL cholesterol levels. And for that reason, PCSK9 is a popular drug target in the pharmaceutical industry for treating high cholesterol levels. So again, we went in, we looked at DNase-Seq data for this PCSK9 gene, and we found the hypersensitive site at the target promoter and made a variety of guide RNAs targeted there. And then we screened these guide RNAs in mouse hepatocytes in culture to see which ones would lead to robust gene silencing. And we can see down at the bottom that we found several that led to very significant silencing by several orders of magnitude. So we then took this dead Cas9 crab fusion protein and corresponding guide RNA, packaged it into an AAV vector that we could, in this case, we could deliver systemically, and then most of the virus will go to the liver and silence gene expression there. But I guess I should point out that, you know, the same type of AAV delivery tools could be used in the brain for the types of targets that are more of interest to this audience. But what we saw in this study was after a single administration of the virus, we could look out over, in this case, 6 weeks and see very potent silencing of PCSK9 expression, looking at PCSK9 protein levels in the serum. But this experiment was out to 6 weeks, but we actually have taken some of these mice out to 6 months. And again, seen after a single administration that we continue to get silencing of this target gene, and that the decrease in PCSK9 levels in the liver correspond to a decrease in cholesterol levels as well, about a 30 to 40% decrease in LDL cholesterol levels. So I'm highlighting this just as an example of how the kind of whatever we're finding with using these tools in cell culture can also be expanded to looking for functional and phenotypic effects by modulating gene expression in vivo. So all of that work was using these targeted repressors or these CRISPRI tools. But I think another exciting use of this kind of dead Cas9 epigenome editing technology is for gain of function by making targeted activators. And that's, of course, because there's lots of tools available for inhibiting genes or products of genes, whether it's antibodies or drugs or using RNAi to repress target genes. But there's actually relatively fewer tools for actual gain of function, especially when you're talking about endogenous enhancers or target genes in the genome. So to make kind of targeted synthetic transcriptional activators, one of the first things that we did, alongside many others in the field who have done similar work, is to attach these VP64 activation domains to dead Cas9. So this is a synthetic peptide derived from a viral transcription factor that will recruit different components of the transcription pre-initiation complex and activate target gene expression. And we've put this synthetic peptide either on the N terminus or the C terminus or both ends of this dead Cas9 protein and then look to see whether we can go and activate target gene expression. And this was one of the first experiments we did. We took a combination of guide RNAs, targeted to a gene promoter, in this case IL-1RN, that we know is off in these cells, and showed that we could get robust activation of that target gene. Now, of course, we had similar questions about the specificity of these tools. So you put this very robust activator and overexpress it in these cells. Sure, the target gene went on, but what else might be getting changed across the genome? But again, when we did RNA-seq, we were pleasantly surprised to see that in this particular experiment, the only thing that changed genome-wide was the 4 isoforms of the IL-1RN gene as shown here. So this gave us confidence that this could be used for a very precise gain-of-function tool to go and activate different sites in the genome. Now, another question that very commonly comes up is how, what's going on with these synthetic activators being able to access these silent and often heterochromatic regions of the genome to activate these silent genes. And so to look at it a little bit more carefully, we first wanted to see what are all the components that we need to turn on the target gene. And so this experiment here shows that, in fact, you need the dead Cas9 and the VP64 activator and the guide RNA in order to get activation of this target promoter in the genome. But then again, with Greg Crawford, we used DNase-Seq to find out what's going on with chromatin state in these cells. And so if you look in the middle, you see where we've targeted these guide RNAs to the IL-1RN promoter. And the first 3 tracks at the top show either with kind of no Cas9 or with different, either Cas9, dCas9 alone or the dCas9 activator, but using the first 3 tracks show guide RNAs that are targeted to other sites in the genome, we get no changes to the IL-1RN gene. But the two tracks in red at the bottom show that when we use the guide RNAs targeted to IL-1RN promoter, we get increases in chromatin accessibility, either with dead Cas9 or with the dead Cas9 activator. So this implies that these dead Cas9 or dead Cas9 fusions are able to go in and access these sites and displace nucleosomes. And when we quantify this across biologic replicates on the right, we see that the increase in accessibility is really the same, whether it's dead Cas9 alone or dead Cas9 activator, even though I showed on the previous slide that the activator domain is necessary to get corresponding gene induction of gene expression. And so we thought this was interesting because it basically suggests that these tools could be used for independently controlling chromatin remodeling and gene activation. But it also shows that these sites are able to get into these regions of close chromatin to activate target gene expression. So all of those experiments kind of gave us confidence that we could use these tools to go in and target different sites in the genome to understand their function. And I wanted to give an example of how we're using this to go in and understand gene regulation and how that dictates different cell phenotypes. And one of our main areas of interest is in using this to better understand cell fate specification. So this is founded on this idea represented by the famous Waddington landscape, where this marble at the top of the hill represents a multipotent or totipotent cell, pluripotent cell that could differentiate into multiple different cell types. And that's represented by it traversing down the landscape and falling into these different valleys, where each valley represents a different cell type. And the hills in between the valleys represent the epigenetic barriers that prevent a neuron from spontaneously deciding to become a cardiomyocyte. And so I think conventionally, we kind of thought that each of these valleys represented a terminally differentiated cell type that it was an irreversible process. But we know now from the reprogramming field and the work of Yamanaka and others that, in fact, these epigenetic barriers can be overcome by activating the corresponding gene network. So, for example, these terminally differentiated cells can be reprogrammed all the way up the hill to pluripotency by overexpression of the Yamanaka transcription factors. Or alternatively, you can kind of directly traverse the hills to go from one valley to another by direct conversion that's been shown for a number of different cell types, again, by overexpression of kind of master transcription factors. So most of these reprogramming or direct conversion protocols do involve overexpression of these master transcription factors. For example, reprogramming the pluripotency or iPS cells with the Yamanaka factors will typically involve overexpression of transcription factors like OCT4. And what we know from a number of studies is that when you overexpress those master factors, they go to many different sites across the genome and thousands to tens of thousands of sites. And in fact, in the typical reprogramming protocols, where you're overexpressing these factors at very high levels, much higher than they would normally be expressed in the corresponding cell type, they will actually go to many sites that you wouldn't normally find them at. So for example, OCT4 during reprogramming will go to sites where you normally would not find it in pluripotent cells. And we also know that binding of those factors to those kind of off-target sites is an important factor in getting efficient reprogramming by going in and loosening up the chromatin and allowing the genome to be reprogrammed to this new state. So in contrast to these conventional protocols for reprogramming cell phenotypes that involve overexpressing of transcription factors that go to many different sites across the genome, now with these dead Cas9-based epigenome editing tools, you have the ability to say, what is the minimum epigenetic perturbation that you need to make in order to alter these different cell phenotypes? Or alternatively, what are the different epigenetics marks that need to change at specific sites in the genome that are sufficient and necessary for converting or redirecting cell phenotypes? And we've been looking at a number of different cell differentiation and reprogramming protocols to kind of better understand this. And one of the model systems that we've been working on is based on this work that Marius Vernig did several years ago, showing that fibroblasts could be converted directly into neurons, either in mouse and then later in human cells, by overexpression of these three master transcription factors, neuronal transcription factors, BRN2, ASCL1, MIT1L, that he calls the BAM factors for short. Now, we wanted to ask the question of, rather than overexpressing these three transcription factors, well, they'll then go to tens of thousands of sites across the genome and have many different effects, can we make a more specific perturbation to the epigenome and to gene regulation to get a similar conversion of cell fate? And so, as a proof of principle, we said, rather than overexpressing the factors, can we send these activators to these target gene promoters in fibroblasts where they're in this repressive heterochromatic state, because of course these neuronal transcription factors won't be expressed in this cell type, and can we reawaken those promoters, activate their expression, and then most importantly, can the activators, these synthetic Cas9 activators, then go away and then we still maintain that activated gene expression state? And the way we're doing that is by using experiments where we use transient plasmid transfection, where we know that there's a burst of expression, but then after a couple of days, that plasmid is diluted out and degraded, and the Cas9 activator goes away. And so for these experiments, we've got three different conditions where we started, in this case, with mouse embryonic fibroblasts, and then we transfected them transiently with either control plasmid. In blue, we've got the plasmids that encode the cDNA, encoding these BAM transcription factors. So this would be the conventional protocol where you're overexpressing these factors. And then in red, I'm showing the CRISPR activators targeted to the corresponding promoters of these genes in the chromosomal DNA. And so when we use qRT-PCR to look at the total expression of these factors in these cells with these different treatments, of course, there's no expression in the control samples because these transcription factors are not expressed in fibroblasts. In blue, you see there's very high levels when we overexpress them from plasmid, and that's what you would expect when you kind of blast these cells with very high levels of plasmid transfection. But then also in red, we can activate the expression of these genes from the genome. Now, it's not nearly as high as what you get when you blast the cells with plasmid cDNA, and the y-axis here is a log scale, so these are large differences in expression, but nonetheless, we could activate the expression of the genes. Now, this is looking at total expression. If we use PCR primers that only amplify the mRNA that's made from the chromosomal copy of the gene, the endogenous copy, by using primers that binds to the UTR sequences, what we see is everything in red on the left-hand side is replicated over on the right-hand side, because when you're using the CRISPR activators, the only place where expression of these genes can come from is the endogenous chromosomal copy. But what you see in blue is that while there's very high levels of total expression of these activators, they're doing a very bad job of activating the corresponding copy in the genome. So there's very low expression coming from the chromosomal DNA. And so we hypothesized that what that would lead to is, since it's a transient transfection, after the plasmid goes away, that we would get higher and more stable levels of conversion into neuronal cells because we've activated the chromosomal copy. And in fact, that turns out to be true. When we look at we mark these cells for actual neuronal markers and neuronal morphologies, as shown in the bottom left, these TUJ1 positive cells, we got about a doubling in the number of cells that convert and express these neuronal markers. So this kind of supports this hypothesis that activating the chromosomal copy leads to kind of more robust and deterministic reprogramming of these cells. These are, of course, immature. They have the electrophysiological properties of immature neurons, and so there's still work that needs to be done to help with the differentiation of these cells. But in order to kind of further investigate the hypothesis that this is because of epigenetic modifications at the target genes and the promoters, we did ChIP-qPCR to look at two active histone marks, H3K27 acetylation and H3K4 trimethylation, at these target promoters. We looked at multiple genes, but I'm only showing ASCL1 here. And at the top, we see the ChIP-seq tracks in developing brain tissue, where these, in yellow, where these marks are enriched, since these are neurons that express these genes. And in blue are the corresponding tracks in the fibroblasts that we started with, where you see that there's no enrichment for these histone marks, given that these genes are off. And then what we see is only with the CRISPR activator treatment do we get enrichment of these marks. This is just looking at 3 days post-transfection of the plasmid cDNA. So it's very fast and robust changes to epigenetic states at these target promoters to activate these genes, whereas you see overexpression of the cDNAs for these transcription factors has no corresponding effect on the epigenetic marks, at least at these early time points after a transient transfection. So this kind of, again, supports the idea that by going in and making very specific targeted epigenetic changes to the genome, we can get more deterministic changes to cell phenotypes. Now, of course, the question would be, how close do these cells actually mimic real neuronal cells that you would get from a brain? And so now it turns out, though, that was a question that we wanted to answer, but was difficult to do, at least at this time in these samples, because as shown in the bottom left, we're still getting relatively low efficiencies of conversion to neuronal cells. In this case, we only got about 4% of these cells expressing this pan-neuronal marker. So to better understand, using kind of genome-wide assays, how well these reprogrammed cell states recapitulate what you would find in a primary cell, we had to move to a more efficient reprogramming protocol. And so to test this, we moved to something we knew would be more efficient, which is the conversion of, or the differentiation of human-induced pluripotent stem cells into myoblasts by activating the expression of this MyoD transcription factor that is the corresponding master transcription factor of myogenic differentiation. So it's the same type of experiment where we use CRISPR activators or MyoD overexpress, well, CRISPR activators to go and activate this repressed MyoD gene in these pluripotent stem cells and look for its activation and induction into a myoblast-like phenotype. And indeed, it was much more efficient. So in this case, we see we can turn on MyoD in almost all of these human iPS cells when we put in the Cas9 activators. And we can quantify that, and we see that more than 70% of these cells, and this is after a lentiviral transduction of these cells, more than 70% of the cells that get the Cas9 activator are activating MyoD expression, which is almost as many we get when we just lentivirally deliver the MyoD cDNA. So it's basically telling us that almost all of the cells that we deliver to, we're able to activate MyoD expression. And at the bottom, I'm showing that when we activate MyoD in these pluripotent stem cells and put them in myogenic culture, we were able to get cells that look like myocytes, they express myocyte markers, they fuse like myocytes are supposed to. This is simply showing that they activate the expression of all the myogenic gene targets. They get enrichment of the active histone marks, just like I showed for the neuronal differentiation. But what I really wanted to show was what do these samples look like when we assay their gene expression and chromatin state genome-wide. So again, to compare the dead Cas9 activation of MyoD versus the MyoD cDNA overexpression, we did RNA-seq to look at gene expression, and so that's shown at the top, and the DNA-seq to look at genome structures across the bottom. And on the left, we have the dead Cas9 activation. So for RNA-seq, we see we get about a little over 600 transcripts upregulated, a little over 200 downregulated. So almost 1,000 total differentially expressed genes when we activate MyoD from the copy of the gene that's in the chromosomal DNA. Now, when we look to the right, we see that if we just overexpress MyoD, we get closer to 3,000 genes that are differentially expressed. So about 3 times more genes that change their expression when we overexpress MyoD than when we activate its expression in the genome. And then at the bottom, we see with dCas9 activation, we get about 3,000, almost 3,000 regions of the genome where chromatin accessibility changes either up or down. And then on the right, it's almost 30,000 regions that change. So almost tenfold to increase in the number of changes in chromatin accessibility across the genome when we overexpress a transcription factor rather than activate it from the genome. Now, the question then is this tenfold increase, this extra 27,000 sites across the genome that are changing, it's possible that those are sites that are supposed to be changed to generate a chromatin state that looks like real myoblasts, which would imply that the dead Cas9 activation was inadequate in generating a myoblast-like genomic structure. Or alternatively, this overexpression of MyoD might be making many different changes across the genome that are inappropriate and don't belong in or don't replicate what happens in a myoblast-like genome. And so to kind of better understand that, we did this analysis where we took DNase-Seq data for 6 different conditions and to look at all of the peaks across these 6 conditions. The first one is the control iPSCs that we started with. Those same iPSCs cultured in myogenic media. Those iPSCs where we had activated the MyoD gene with CRISPR activators, the cells where we had overexpressed the MyoD cDNA, or the last 2 samples are DNase-Seq data from primary human myoblasts and myocytes. And we looked at all of the peaks called across all these samples, and we categorized the peaks based on whether which treatments they were shared across. And so that's the bar graph shown here, where the 0s and 1s indicate the classes of peaks that were shared across those samples. So if we look all the way to the right, at the second to last bar, you see that this bar corresponds to all 1s. So those would be peaks that are shared across all samples. And so those are, you would kind of expect a lot of those types of peaks because those are probably peaks that are corresponding with like housekeeping genes. So genes that are on in all cells or regions of the genome that are open or in all cells in order to keep the cells healthy. And so it makes sense that there's a lot of those peaks. But what's striking is that the last bar, even more than those peaks that are shared across all genes, is 0, 0, 0, 1, 0, 0. So those are peaks that correspond, or those are peaks that only exist in the samples where there was MyoD cDNA overexpression, and those peaks are not shared across any other sample type. So that indicates to us that these are peaks that are really aberrant, inappropriate peaks that don't belong in any of these other cell types and are the result of kind of massive overexpression of this master transcription factor that is probably going to many other sites in the genome and causing effects and opening of chromatin that don't belong there. And so this is just more evidence to us that these CRISPR-based activators can be used to give more specific and kind of natural types of changes to the epigenome than kind of the other types of conventional protocols that are out there. And then in the last couple of minutes, I'd say everything that I showed there was with these kind of genetic or generic activators and repressor domains that kind of make many different changes to the epigenome or gene expression in parallel. But we've also been wanting to use these tools to go in and study the function of very specific epigenetic marks. So for example, that VP64 activation domain will recruit lots of different factors that cause multiple epigenetic changes in parallel. So to better understand how specific epigenetic marks change gene expression and chromatin states, we've been attaching different enzymes to dead Cas9 that modify a narrower set of changes to the epigenome. And so one of the first examples of this, although we've done many others now, was fusing this P300 acetyltransferase to dead Cas9. So at the top, we've got the kind of conventional fusion, or the first fusion that we made, which is that VP64 activator. Then we took the full P300 acetyltransferase and attached that to dead Cas9. And then we had another fusion that was just the core domain, the catalytic domain, and did not include the N and C terminus of P300 that are known to interact with many other factors in the cell and have different types of autoregulatory function. And so we wanted to understand if we just target this acetyltransferase domain to different sites in the genome, what would be the changes in gene expression. And so what we did is took those different fusions and targeted it to 3 different human promoters in human cells. And what you see first is the dead Cas9 has no effect. the dead Cas9 VP64 has a potent effect in changing gene expression, like I showed in the previous slides. And then disappointingly, that full-length P300 fusion had basically a negligible effect on gene expression. However, when we included the P300 core domain without the N and C termini of P300, we saw a very robust activation of these target gene promoters, even more so than these first generation dCas9 activators. And then we also included an important control that includes this this point mutation to a residue in the catalytic domain that inactivates its function. And then there we see that it's really the acetylation function of this core domain is really important to getting that corresponding gene activation. And then we also used RNA-seq once again to show that, and this is just using this model promoter for IL-1RN, that we get very specific changes to gene expression when we target it to this gene promoter. So now that showed that we could target this acetyltransferase to promoters and activate genes, but of course we're also really interested in what this fusion does at enhancers, given that one of P300's normal function is that it's localized at active enhancers where it catalyzes H3K27 acetylation. And of course, this gets into how we're using this to follow up on GWAS and look at how genetic variation corresponds to gene expression. As many of you know, that the vast majority of polymorphisms identified in GWAS studies fall in these non-coding regions and often in enhancer elements. But up until now, there's been no tools to really go in and directly perturb these enhancer elements, especially for gain of function type studies. And what we observed is, again, in this case, going back to this HS2 model enhancer, is that in fact, if we targeted this dCas9 P300 core domain to HS2, we could get corresponding increases in the expression of its distant target genes, the different globin genes from 10 to 50 different kilobases away. And this was something that the dead Cas9 P300 fusion was uniquely capable of doing, that, as you see here in these blue bars, that the VP64 activator could not do when it was targeted to the enhancer, as opposed to when it was targeted to the promoter, as shown by no effect in the green bars here. So that was showing that we could activate gene expression from the enhancers, but we also wanted to know what was happening at the epigenetic to the epigenetic marks when we targeted enhancers. So this is looking at H3K27 acetylation enrichment. And so first, on the left here, I'm showing what we saw at the target HS2 enhancer, which is where the guide RNAs were targeting these dCas9 fusions to. And what was interesting is that either VP64 or the P300 core domain, both of these could lead to enrichment of P300 at that, or enrichment of H3K27 acetylation at that target enhancer. Which is, we knew that VP64 recruits endogenous P300, so that wasn't surprising. But what was surprising was that when we target that enhancer, we only get corresponding enrichments of H3K27 acetylation at the target promoter when we use the P300 fusion, and we don't see that with VP64. So, so that indicated to us that this P300 fusion might be a unique tool for going in and getting gain of function of these target enhancers where we see corresponding effects at target gene expression. So that work demonstrated that we would use those tools to target specific DNA and specific putative regulatory elements and see what their corresponding expression or corresponding effect was on gene expression. But of course, most of these studies have identified, many different putative regulatory elements. And working with our collaborator, Greg Crawford at Duke, he's identified hundreds of thousands of sites in any particular cell type and millions of sites across all human cell types that show this kind of open chromatin state and may be important in regulating gene expression or different phenotypes. So in order to be able to look at these things in more high throughput rather than one at a time, we've been developing CRISPR-based epigenome editing screens to go in and do this work. So many groups are familiar with CRISPR-based screens where they're looking at, for example, gene knockout. We're doing loss of function of coding genes. But what we wanted to do here is use a single guide RNA targeted to a regulatory element to either inhibit its function or activate its function. And so as one example, we started these experiments where we used DNase-Seq data to map out all the putative regulatory elements in a particular region of the genome, although you could imagine using ChIP-seq data or looking for different DNase-Seq or ATAC-seq or ChIP-seq peaks that are differential between two different cell types or cell states. But designing libraries of guide RNAs where the guide RNAs are targeted to all of the DNase-Seq or ChIP-seq peaks in these areas of the non-coding genome, synthesizing all of those guide RNAs on arrays and then cloning them into lentiviral vectors and introducing them to cells where each cell gets a single lentiviral vector, so it gets a single guide RNA, and those cells also have been engineered with either the dCas9 CRAB repressor that I showed can repress regulatory elements, or the Cas9 activator that I showed can activate these elements. So then the effect is that each cell in this population has gotten a loss or has gotten a single perturbation to one regulatory element across the genome. And then you can perform some type of selection of those cells, either based on some type of phenotype or drug selection or growth or death or changes in expression of one or more particular genes, and then select those cells, sequence the guide RNAs in those cells, map them back to the genome, and thereby identify regulatory elements that correspond to that particular gene or phenotype that you're interested in. And so we published that earlier this year, and this is one example where we were looking at expression of this model oncogene HER2. And we actually, and this was a very small screen, but in the screen we looked at 5 megabases of the genome, more than 400 different DNase-Seq peaks, and about 12,000 different guide RNAs in a single pooled screen. And so we're just looking at a very small region of the genome that we screened, right around the target gene for which we were selecting based on expression of this gene. And you see that we get hits at the promoter of this ERBB2 gene that encodes HER2, also some intronic enhancers, but we also found a number of hits at distant regions across the genome that we're going in and characterizing in more detail now. And just to point out a couple of other interesting aspects of how this tool or this technology can be used to screen putative regulatory elements or GWAS hits across the genome. One of the interesting things was to look at this across different cell types. So here we did the same screen in 2 different cell types, A431s and 293Ts. And if we just look, so the tracks for these 2 different cell types are shown on the right in panels B and C. But on the left in panel A, we're plotting all of the enriched or depleted guide RNAs for A431 cells on the x-axis or on the y-axis, and for 293 cells on the x-axis. And what you see in yellow, there's many shared hits, but you see in blue and green, there's also many hits that are specific to that particular cell type. So doing the screen in 2 different cell types allowed us to basically find what we think are cell type specific regulatory elements for these different genes. And then similarly, we can do the exact same screen with the same guide RNA pool, but either with the P300 gain of function approach or the CRAB loss of function approach. And so again, panel B here shows the gain of function with P300, panel C shows what we see with loss of function with the CRAB domain, but the panel on the left shows that if we graph the P300 hits versus the CRAB hits, in the top left quadrant in yellow, we see all of the hits that are shared for both of those, but in blue and purple, we see the hits that are unique to the gain of function or loss of function approach. And that is something you would expect, because you might say that in a cell type where the gene is already, where a regulatory element is already on, you will not maybe get those hits for a gain of function screen, but you will get those for a loss of function screen. Alternatively, for a gene that's off, you might not find regulatory elements with a loss of function screen, but you would find those regulatory elements with a gain of function screen. So this is the type of approach that we want to use to go in and screen all of the different regulatory elements that are associated with some of these different GWAS hits to follow up those that Patrick and the PGC have discovered, but still need to really understand what their effects are on corresponding gene expression. So that's all of the slides that I wanted to show, but I'm happy to answer questions. Just to summarize, I think that my impression is that the general community is more familiar with CRISPR as a gene editing tool if you want to make specific gene or genome sequence changes. But what I wanted to do here was highlight how it can be used for a number of different areas in changing genome structure and function in surprisingly highly specific ways, and then how we can adapt that for high throughput screens of non-coding regions, either associated with different phenotypes or identified by GWAS. I'll just point out, I mentioned each of the students and postdocs in my lab along the way that have done the work, but all of this epigenome editing, and especially the functional epigenomics work, has really been the result of a close collaboration with both Tim Reddy and Greg Crawford here at Duke. And if there's time for questions, I'm happy to take them.
Excellent. Charlie, thank you so much. There's a fascinating talk about these increasingly precise and scalable tools that you're coming with. For the participants, you can ask a question in the Q&A box or in the chat box, and we'll field them as they come. Charlie, I had a couple of questions to get started, if I could, please.
Sure.
First, the direct conversion from fibroblast to neuron stuff is impressive. But a major issue in the field is how to get a more mature and a more functional neuron faster. Because typically, you have to have these things in culture for a very long time. I know of people that have been, looking at cultures for years, in fact. Have you looked at this at all? Is there any sort of tricks that could be where you could hack a cell to actually make it more mature more quickly?
So it's certainly not a problem that we have solved yet, but it's something we're very interested in and that we're kind of working on actively. So from a big picture, like blue sky perspective, where we're going with these tools is, conceptually, if we can use these tools to modify any epigenetic state in the genome in any cell type, then you would imagine that if you can define a specific epigenetic state that you want to create, then if these technologies get robust enough, you should be able to go in and create that in any starting cell type. So where we're going with that is saying that if you can characterize using ChIP-seq and other tools what the epigenome looks like in those more mature neurons, then we might be able to use these technologies to go in and create that epigenetic state and presumably the corresponding more mature phenotype with these types of tools, rather than the kind of the more conventional approaches of saying, what are the 2 or 3 or 4 transcription factors that we think are associated with this state, and then carpet bombing the genome with those factors. So
got it.
So it's something we're actively working on, where we're hoping we can more deterministically create these more mature epigenetic states.
Follow-up question, has anyone done a RNA-seq experiment where they've actually followed iPSCs from differentiation through NPC through, aging out the neurons? Has that been done? I mean, surely there are data sets where people have looked at this.
Yeah, I believe there are data sets. I don't know all of it off the top of my head, but that type of work is out there. And so what we're trying to do is say, if we get the most mature, if we get, like for example, ChIP-seq data or TAC-seq data on the most mature neuronal subtypes that we're interested in, what do we need to do to be able to go in and create those? And that's what we're working on now.
Okay. There's no other questions quite yet. Let me come up with the second one, please. So again, as I think I've mentioned to you, the single locus stuff that you've done is compelling about, using the tools you're developing in a flexible way to silence or enhance the expression of very specific genes. What are the prospects for scaling this to large regions or even genome-wide? You touched this in the last few questions, but what technical issues or key issues need to be solved for this to become something that could be done practically?
Yeah, so the big issue is getting coverage of all those sites and getting sufficient cell numbers. So, you can design and synthesize as many guide RNAs as you want. But so, for example, in any one cell type, Greg has shown that there's roughly 100,000 DNase hypersensitive sites. So if you wanted to screen all of those sites, to answer your genome-wide question, and let's say that you wanted to do it as bare bones as possible, and you only did 10 guide RNAs per DNase hypersensitive site, then that's 1 million guide RNAs. And again, if you did this as scaled down as possible, and you only got 100-fold coverage of your library, which is less than you would typically want, then that's 100 million cells. And then there's some technical reasons why you need to get, for like lentiviral transduction efficiency, et cetera, where you would need at least a 5-fold increase over that. So doing experiments with 500 million cells at a time per replicate, that becomes the bottleneck. And so for some cell types, you can do that type of large scale experiment. And that's what we're doing now for example, some suspension cell lines. But for a lot of things, that's problematic. And so for that reason, you just might want to use different criteria to be more specific about which sites you're going to look at. Because certainly doing maybe not 100,000 sites, but 1,000 or 10,000 elements or enhancers is possible. You just need to use different criteria to focus in on which ones you want to look at.
Do you think, given that you're probably going to do this at pretty high scale, do you think there might be rules here? I mean, if you were to get a data set where you actually did a very large number of open chromatin sites, where you silenced them efficiently, and then actually looked at the changes in expression genome-wide, do you think it's possible that you could apply ML approaches to try to understand what are the features of the ones you need, what are the subset you really need to focus on?
Yeah, that's our hope, and that's what we're going to try and do. So we, like I mentioned, in some experiments, we are trying to basically characterize all hypersensitive sites or all possible regulatory elements across the genome, and then find out what all their contributions are, and then look to see if there's rules about which elements are doing what, especially if we try and basically do a functional interrogation of all of those sites using these types of screens, and then overlay that on all the ENCODE data or other epigenetic mark data that we can find to see, can we characterize how certain classes of regulatory elements act in certain ways. So that's definitely one of our big goals.
Makes sense. Okay, there's no questions that I'm seeing from the group, but I just wanted to, and we're past time anyway. Charlie, this has been absolutely fantastic. Thank you so much for taking the time to talk about these exceptional innovations that you guys are coming up with. And,
yeah, no problem. Thanks for the opportunity.
Thank you very much. And that's it for today. This call, the recording will be posted on the PGC site if people want to review it later. But again, Charlie, thank you so much for your time.
All right, thank you.
That's it, everybody. Thanks.